All right, it's a real pleasure of mine to introduce a great scientist and friend to a lot of us, Dr. Tom Waltzek. He earned a BS from Florida State University in marine biology, and then went to the University of California, Davis, where he earned an MS working on functional anatomy and ecology of cichlid fishes. He continued to study in a dual degree DVM PhD program at UC Davis, focusing on fish health, where he earned his DVM in 2009 and finished his PhD dissertation on the evolution and ecology of viral diseases of aquatic vertebrates in 2010. After graduate school, Dr. Waltzek moved back to Florida as a postdoc and eventually a faculty member at the University of Florida's College of Veterinary Medicine. While at University of Florida, his research focused on emerging pathogens impacting both aquatic wildlife and aquatic animal industries. And more recently, in 2021, he joined the USDA aquaculture trade staff in support of the National Aquaculture Health Plan and Standards. So thank you again, Tom, for presenting today. We're really looking forward to your talk.
Awesome. Thanks, Nick. Thanks, Matt and Megan, for inviting me to speak today. I was excited to give this talk because, as Nick was kind of describing a bit of my background in academia first and now working at USDA, it allows me to kind of combine some of the things that I find really interesting from the research and diagnostic front that I did in academia on tilapia lake virus. And then more recently, since I've been wearing more of a regulatory hat at USDA, talk about how sometimes research and diagnostics impacts not U.S. aquaculture through trade regulations. So I'm hoping this talk will give you a little bit of information about the disease, research that's been done, even future directions for tilapia lake virus, but also tell you a little bit about, again, how research and diagnostics impact U.S. aquaculture through trade regulations. So it'll be kind of a mismatch or a matchup of diagnostics and research and regulations in the United States. Okay. All right, so Nick did a good job of introducing myself, so I'll skip that part and just go straight into tilapia aquaculture. Of course, tilapia is globally produced, it's a globally produced species, and it's the second most important aquaculture species after carp species around the world. So super important as a protein source. In many parts of the world, it is the primary protein source. Global annual production is estimated around $11 billion. And again, there's many different tilapia species that are raised and hybrids and so on and so on. And the tilapia aquaculture has become quite sophisticated, and in some regions of the world and in other places, it's just simple ponds or cages and things. So in the United States, we have around 200 farms, and we're about 6% of the global market. And tilapia are a relatively disease-resistant species. They grow well. They don't have, extensive husbandry needs that are different from other species. They grow fast and things like that. And overall, even resistant to some species, to many different diseases and such. Those of you that work with tilapia will know that bacteria is probably the primary problem that are seen from the infectious disease standpoint. Things like Streptococcus species, Francisella, many other types of gram-negative and gram-positive bacteria and such. We have water molds, things like Sapolignia and such, and of course parasites are true across fish species as a problem. But again, bacteria are really one of the big problems in aquaculture of tilapia species. As far as DNA viruses are concerned, there are a few viruses that have impacted tilapia aquaculture. Iridoviruses have become a pretty important problem globally, and that's infectious spleen and kidney necrosis virus, which is a megalocytovirus. I won't be talking about that today, but that's emerged as a pretty important problem. And there's reports of herpes viruses, and parvovirus has recently been detected by one of our Thai colleagues, Dr. Winsar Chetpong, in tilapia as well. But The most important viral disease that's emerged is tilapia lake virus. So I'll talk a little bit about tilapia lake virus and continue talking about the research and diagnostics now. So what is this disease? Tilapia lake virus disease, it first was reported in Israel aquaculture in 2009. It causes a lethal systemic disease, meaning affecting all the organs of the fish. Internal and external lesions are seen. And it reports in significant mortality, anything from 20 to 90%. And I'm going to use I'm not sure, can you guys see the arrow if I use it?
Yes, we can.
Okay, great. So you can see these external lesions here, some bleeding that you can appreciate. And internally, we often see swelling of the internal organs and ascites or fluid within the coelomic cavity. And it's pretty non-specific in the clinical signs. So the fish will go off, they won't feed. They'll be lethargic and kind of swimming abnormally. That's very common, and they'll be laying on their side before they die. I've talked a little bit about the gross lesions already, but often they'll be darkened in appearance. They may have bulging eyes, exophthalmia, skin and fin ulcerations, and the ascites, scale loss, and even pale gills and such. From a histopathology standpoint, a very interesting lesion that's seen primarily in the liver is a syncytial hepatitis. And so syncytia is something that we think of with viral infections, and what a syncytium is a series of cells that have fused and they become multinucleated, because as they fuse, they accumulate all of the different nuclei in one giant cell. And there are other things that can cause syncytium, but viruses are one of the most common causes of syncytia. Herpes viruses cause syncytia, Rio viruses cause syncytia, and tilapia lake virus causes syncytia within the liver. So the liver is one of the best organs to sample, and it's definitely one of the hardest hit organs with this disease. It also goes to the brain. We see an encephalitis with lymphocytes and perivascular cuffing. So we see an inflammatory response around the vessels in the brain. And we see focal hemorrhages and congestion of blood vessels within the meninges, the surface layer of the brain. So that's a little bit about the disease and what it looks like grossly and microscopically. So it's a novel orthomyxovirus-like. So when we think of orthomyxoviruses, right, we're thinking about flu-like viruses. Infectious salmon anemia virus would be the classic example in fish, and it was thought to perhaps belong to that family initially, but after it was sequenced, it was divergent enough from the orthomyxoviruses to actually become its own family. And of course, within a new family, it's the monotypic or only genus, and so on and so on. So this is actually a new family of viruses and a new genus of viruses, of course. It's a negative sense single-stranded RNA virus with 10 segments, a pleomorphic virion. So it's kind of roundish, but sometimes pleomorphic virus particle, 50 to 100 nanometers in size. And then how do we make a diagnosis? Well, it grows relatively well on the striped snakehead cell line, the SSN1 cell line, at 25 degrees Celsius is the optimal temperature. And it can grow up to about 10 to the 6th TCID 50 per mil in that cell line. And there are other cell lines that are permissive to tilapia lake virus as well. Some of the tilapia cell lines are also permissive to it, but it grows relatively well in cell culture. And now there are various molecular diagnostics, from conventional RT-PCRs to quantitative PCRs and situ hybridization. And as time has gone on, now we have LAMP assays and quite a few other molecular techniques for the diagnosis of tilapia lake virus. When I started doing research on tilapia lake virus, there was a serious need for validated diagnostic assays, and I'm going to tell you a little bit about that. But let's hold on to that thought. I'll come back to the need for validated diagnostic assays and some of the research that we did. So again, in cell culture, here you have a panel of cells, SSN1 cells that are okay. And then you can see some unhappy kind of refractile cells that are pulling up and dying here, leading to these open areas or these plaques within the SSN1 cell line. So From a diagnostic standpoint, grows in cell culture. We have great molecular assays. Even electron microscopy can be used to help with the diagnosis. It is globally distributed at this point, 15 countries, 4 continents. As I mentioned, it was first found in Israel and about the same time in Ecuador by Dr. Hugh Ferguson and colleagues. And then it appeared in Asia, Africa, and then in South America. And in 2019, we had the first detection in North America that I'm going to tell you a little bit about. So we actually have quite a few countries now that have at least reported cases on 4 different continents. Globally distributed disease and a problem in tilapia aquaculture in many of these areas. So what about the emergence of TLIV in the United States in 2019? That was in our Nile tilapia industry. Well, and one of the points before I get into this story that I want to make sure I hammer home is the importance of collaboration. And without the collaboration of our private laboratories that I'll be talking about, our university laboratories, and our national veterinary services laboratory, all of the story that I'm going to tell you couldn't have happened. And so that will be something that I really hope for the younger members that are listening to this talk to think about your future and the importance of collaboration. So the story starts with a private lab, Kennebec River Biosciences. Dr. Bill Kelleher made a detection of a pathogen coming in from imports of Nile tilapia from Thailand. And that was in cell culture. So he was growing something in cell culture from an import of Nile tilapia from Thailand. And he had something in cell culture that looked like I described to you, plaque-forming agent, and he wasn't sure exactly what it was, but he immediately sent samples to my former laboratory at the University of Florida, and he also sent it to the National Veterinary Services Laboratory as well for confirmation. And so we developed a qPCR based on work that we were trying to do from a research standpoint. And so the first detection happened in February of 2019. You know, he sent it to us 2 days later. We were able to tell him by qPCR, look, this is tilapia lake virus, as he had suspected. Shortly thereafter, it was also confirmed by RT-PCR by NVSL, and they did gene sequencing to also show that it was essentially tilapia lake virus. We then went on to collaborate with Bill, sequenced the genome for 2 different isolates he'd gotten from the United States and published that to prove that it was going on, that it was tilapia lake virus in the first detection in the United States. The other thing I wanted to mention is before the facility that he had received samples from in the United States, that facility had received tilapia from Thailand. And they didn't have outbreaks until they had that import from Thailand. So they started losing fish that were commingling with the Thailand fish. That's what started all this. They were having an outbreak. They sent the samples to Kennebec. He made that detection, and that's where the story kind of unfolds as I just described. But before that, they had actually moved fish out of Idaho and into Wyoming and Colorado. And so the outbreak continued to spread outside of that one facility into additional facilities in different states. And I want to make the point now, because we were able to make that diagnosis, they were able to contain, to the best of our knowledge, this disease in those facilities before those facilities shipped all over the country and spread that and let this virus become endemic in the United States. And so as a result of that, you know, those facilities were put under quarantine, depopulations were put into place, and that was all work done by USDA, as I'll describe. But again, the importance of our various laboratories, whether or not it be private, university, or in our national laboratory, the importance of them working together, I think, is illustrated by this story. And one of the other messages here is that we do not think we have tilapia lake virus in our Nile tilapia industry in the United States. And one of the reasons is because we're actually doing diagnostics, developing diagnostics and working together. And I don't mean to make it sound like that there was a seamless passing of the baton in this story, but it was functional enough that we, again, think we prevented it from spreading and becoming endemic in our country, the way it is in many other countries, especially in Asia. All right, so I've talked to you about TLIV being a global threat to the tilapia aquaculture industry. What I've told you is that through collaborating U.S. laboratories, we were able to rapidly make a diagnosis following the importation of tilapia in 2019. The USDA response was to quarantine those facilities, depopulation, clean and disinfect those facilities before they received more Nile tilapia, and that contained the outbreak to the best of our knowledge. And that work was done by my colleagues at USDA, our commodity, or folks that deal with domestic issues on the home front, so to speak. Dr. Nancy Hanaway, Kathleen Hartman, and Lynn Creekmore were all a part of that. And that was a very important contribution to this response based on that outbreak. Again, I mentioned that Dr. Hassan and many of our collaborators then published the first genomes of these viruses on those isolates that came from Thailand. All right, so we talked a little bit, what about the impact and our USDA response? So we'd had a detection. We knew that there was a potential risk now for seeing tilapia lake virus again from our imports. What could USDA do about it, right? So that was diagnosticians working together, but what can USDA do to close the gates, right? What can we do to close the gates and prevent more outbreaks of tilapia lake virus coming in from our imports from overseas and such? So within USDA, there's many units. One of them I've talked about already, commodity, but we also have an epidemiology unit, and the CIA, or Center for Epidemiology and Animal Health group, performed 2 different risk assessments to understand the risk pathways. How might the virus be introduced? Well, one pathway is obvious and is what happened in the only known outbreak of tilapia lake virus in the United States, and that was the importation of live fingerlings. So their risk assessments, the 2 separate risk assessments that they did, showed that obviously importing live tilapia fingerlings poses a high risk of seeing this disease again. They also were able to analyze the potential of frozen tilapia fillets to introduce tilapia lake virus, and it was assessed at a low risk because tilapia fillets are unlikely to encounter or come in contact with the tilapia aquaculture industry. Market size tilapia that are imported from Canada and destined for our domestic terminal live markets, like wet markets that you might see in San Francisco or Chicago, where there's tilapia in tanks that are coming from Canada as full-grown fish. Somebody points to them and they're essentially euthanized, taken home and eaten. That's another market that was of risk, but it's considered a low risk because those wet markets are also believed to be not linked epidemiologically to our aquaculture industry. So the high risk is live tilapia fingerlings being imported into the United States, and that was done. So essentially, the risk is live animals, and that's the greatest risk to the United States. And so with that information, a federal order was put into place. So USDA went into action in 2019, the same year that we actually made the diagnosis, to put in import regulations to close those gates on bringing in tilapia that are not known to be free of tilapia lake virus. So a federal order went in place, and that impacted all US imports of live tilapia, fertilized eggs, and gametes from TLIV susceptible species, that primarily being tilapia, various tilapia species. So all live tilapia had to actually have several regulations and testing occur before they were allowed into the United States. So it was issued in accordance with the regulatory authority provided under the Aquatic Animal Health Protection Act to USDA. The purpose, of course, is to prevent the entry or introduction of TLIV into the United States by restricting imports from all countries. And it applies to TLIV susceptible species, Nile tilapia, commercial hybrid species, red tilapia, wild tilapia. And it requires the following: all shipments are accompanied by a USDA import permit, a health certificate endorsed by an official veterinarian or an aquatic animal health official of the exporting country's competent authority, and that it's undergone veterinary inspection at one of the US designated ports. Okay, so we put in regulations to hopefully stop this disease from coming in on our imports again. And currently where this sits is a federal order is a temporary fix. It's a patch, and it is true law, but it can't last forever. If you want it to become true law and become permanent US law, we have the Code of Federal Regulations, which we call the CFR. That's this kind of red covered book here. And Dr. Marston and Dr. Shanna Siegel, they're the directors of our trade in the United States, especially aquatic animal. Dr. Shanna Siegel is our National Director of Trade, and Dr. Alicia Marston is our Aquatic Animal Health Director of Trade. And they're working on actually getting this law codified within the CFR, right? So making this permanent US law. And I just wanted to mention also, you might wonder how many different regulations we have in the United States to protect us from emerging diseases or notifiable diseases in the United States. And we have very few, actually. This is only the second disease for aquatic animals that we have import regulations on. And the other one is spring viremia of carp. So this is only the second disease that we have import regulations for an aquatic animal health or aquatic animal species. So that essentially closes the door on the import of TLIV into the United States. It's not perfect, but it's much better than doing nothing. And that was the response. I'm going to go back just 1 second. I want to make one last point about this. That was the appropriate response as deemed by USDA. And I wanted to mention one last thing. This federal order came out in 2019, at the end of 2019. And the diagnosis was made in basically February, March of 2019. So 6-7 months later, we already had regulations. And so I wanted to make the point, oftentimes we think our government is very slow to respond, inefficient to respond, and perhaps that is the case, but this represents an example where collaborating laboratories made a diagnosis rapidly, and then USDA jumped into action, assessed the risk of this pathogen, understood the risk pathway, and then put in import regulations to stop that potential risk and stop tilapia lake virus from becoming endemic in the United States. So this represents, I think, an example of where collaboration and the US government and USDA acted quickly. And again, I don't mean to make it sound like there was a seamless passing of the baton, many hiccups and so on, but it's encouraging from a person that works in academia and now works at USDA that we can all collaborate together to essentially affect change that helps our aquatic animal health industry within the United States. All right, so now we had import regulations and such, but one of the problems we realized at the same time we were putting import regulations in place was, in fact, we didn't have a well-validated assay for the diagnosis or confirmation of tilapia lake virus. And so at the time, I was still at the University of Florida before I joined USDA, and I'd written several grants in order to develop and validate a TACMAN qPCR assay for TLIV surveillance. So these were USDA grants, NIFA and AFS and other laboratory. We collaborated with many laboratories in order to work together on a reproducibility study that I'll talk about as well, an interlaboratory reproducibility study. The goal was, with validation, was to use the OIE, which is now the World Organization for Animal Health, WOA, was to use their stages of assay validation and design and optimize a tilapia lake virus qPCR and then validate it by the WOA standards. Again, stage 1 through 4. We've gotten through stages 1 through 3 versus using the various, through the various grants that I mentioned and such. So what I want to tell you a little bit about now is a little bit about some of the research that we did and the development of the diagnostic assay for tilapia lake virus, the qPCR assay. So the first step that my group has always taken was, if you want to design a PCR assay, you have to decide what you're trying to do. We were interested in a pan-TLIV qPCR, meaning that it detected all strains of TLIV. The problem was, at the time, we only had 1 or 2 genomes in GenBank to look at. And so I got with the world's foremost expert, in my opinion, on TLIV, Dr. Win Surachetpong from Thailand, a great researcher and somebody who's really led the way, in my opinion, on tilapia lake virus research. And he had isolates from around the world. So we ended up sequencing a bunch of different strains from Thailand, which is where he's from, but he also had Israel, and we also had Ecuador. So we had basically the Middle East, we had Asia, we have Colombia, Ecuador, and so we've got South America, and again, Asia again in Malaysia, and then South America, Peru, and then Africa. The Egypt samples were critical as well. Once we had sequenced the whole genome, then of all of these strains from around the world, we could look for completely invariant regions that have no variation and are also decent primer and probe binding sites for the assay. And what we ended up finding was that segment 10, a 64 base pair region of segment 10, was not only relatively invariant, it does have some primer mismatches in the forward primer and the reverse primer, but there was only one strain in which there's 2 mismatches, and it works great, tested empirically in the laboratory, even against that Egypt strain of tilapia lake virus. Most of the isolates have none or only a single mismatch, and we've tested all of these, and it works well against all of them. And this was the best site. Most sites are definitely much more variable, and so this was both a good region for primers and probe, but also because we had access to all of the genomes, we could design a primer with a minimal number of mismatches to the primers and probes for the TACMAN assay. All right. The next step that we talked about is, once it was initially working in the laboratory, optimized and such, we wanted to look at the analytic performance, right? So the sensitivity and the specificity. So when you think about sensitivity, right, we're thinking about the limit of detection of the assay, the ability of the assay to detect, a small number of copies in a sample. How sensitive is it really? Can it detect 10 copies? Very few assays have problem on the other end, right? Nobody's going to have problem with 1,000 or maybe 500,000 copies of a target. But qPCR, our assays can be challenged if we get down into that 10 copy range. And so that's all about the sensitivity of the assay. We also wanted to know it was specific. So does it detect tilapia lake virus? That's its target, but hopefully it doesn't detect other viruses that are related to it. It should be specific to tilapia lake virus and not cross-react with other agents. So in order to look at this, we did 48 separate experiments, linearized plasmid dilutions. And so we're taking the qPCR target in a plasmid form. We're doing dilutions in which we've got a vial that has either 10 million copies, 10 to the 7, all the way down to 10 viral copies, running all those in triplicate and doing 48 separate experiments of 10 to the 7 to 10 viral copies in triplicate. So we ended up showing that it detected 10 copies in 100% of the reactions. So it definitely detects down to 10 copies without much trouble. And the addition of host RNA has almost no effect on the assay as well. So it still detects down to 10 copies with the addition of tilapia RNA, which is more realistic of a tilapia sample. So the limit of detection of the assay is 10 copies or less. We were also able to show, since we were running it in triplicate, we could look at the variability on the triplicate. So each different sample run 3 times shows us, is there any variation for one particular sample? The intra-assay variability, there was a very low coefficient of variation. We're also able to compare among those various experiments is there variation between experiments, the 48 experiments, and that was also had an acceptable and very low coefficient of variation. It has a slope of about 3.3, close to 100% efficiency, so it had great performance overall, and obviously very sensitive, as we said, so the analytic performance was excellent. And it has no cross-reaction with infectious salmon anemia virus, influenza A or B, beta notaviruses, fathead menopacornavirus, and we even tested some DNA viruses and things as well. So it appears to be specific for tilapia lake virus. So that's the analytic performance. The real problem came to the diagnostic performance, and this is where we had written grants to do challenge studies, because in order to do diagnostic performance, you have to compare your assay, the new assay's performance, to a gold standard or known set of positive and negative samples. Well, how do you know that the samples are positive and negative? Well, because you're using some other assay as a gold standard or some other definition of knowing that they're positive and negative. And now you have to compare your new assay to those known positive and negative samples. But at the point that we got involved, there was no really positive or negative material. There's lots of negative material in the United States, but there wasn't a lot of positive material. So we developed a challenge model by injecting fish, Nile tilapia, in the laboratory with the virus. Since it can be grown in cell culture, it propagates easily in SSN1. We just did simple challenge studies and developed known positive samples. So a fish is injected, it shows the clinical signs. It dies, we do an RNA extraction on the liver. Now we had a known set of positive samples from our challenge studies that we developed. So the optimized qPCR, we had a 91 proven positive samples and 180 proven negative samples. The negative samples were fish that were not challenged with the virus, or that were a part of field samples that we'd had from facilities that have never had outbreaks to our knowledge and such. And so we had 91 known positives and 185 proven negative samples. So the diagnostic sensitivity, again, comparing our assay to known positive and negative samples, was very high. We had a 96.8 diagnostic sensitivity and 100% diagnostic specificity. So that gave us the analytic performance. This was the diagnostic performance. That's the stage 1 and stage 2 set forth by the World Organization for Animal Health on how to validate assays. So we had gone through stage 1 and stage 2 through that grants that I mentioned previously through USDA. And I'd be remiss if I didn't mention our colleagues who worked with us. This is Paul Hick, formerly of the University of Sydney. Now he works at an agency similar to USDA in Australia. And Dr. Lori Gustafson at USDA. She works at SIA. She's a great epidemiologist, a good friend and a colleague that helped with the analysis of our qPCR and helped us think about the epi aspects or epidemiology of what we were doing and assay and the design of the assay and such. The last part of it, as far as we've taken this, is stage 3 of assay validation as set forth by the World Organization for Animal Health. And we worked with NVSL in a small grant that was allotted to Dr. Kudi Subramaniam and myself. This was an interlaboratory reproducibility study. And so we had this assay that we had an SOP for, standard operating procedure for analytic and diagnostic performance. We knew it performed really well in our hands, in our laboratory. But the real question was, how did it perform in other laboratories? So if we gave somebody a known set of positive samples, a known set of negative samples, and we gave them an SOP in which we give, here's the primers, here's the probes, here's the samples, you can run qPCR2, take this assay and this cookbook instruction, and let's compare how various laboratories actually do using the same set of known positive and negative samples. So we had 6 laboratories and we gave them the SOP. We had 31 positive samples and 19 negative samples. Excuse me. And Waddle was one of our state laboratories in Washington State University, also a veterinary laboratory. Our state laboratory in Florida participated, the Bronson Animal Disease Diagnostic Laboratory. Dr. Reddy Baminini was a big help. Kevin Snetbeck at WADA was a big help. Dr. Subramaniam and myself performed this work at a university laboratory. So we've got state labs, we've got university laboratories. Kennebec River Biosciences, a private fee-for-service diagnostic laboratory, participated, which was very nice to have a private lab working with us as well. The New Jersey State Laboratory, and of course, our Janet Warg at our National Veterinary Services Laboratory participated as well. And I don't want to go through a lot of the data for this. We're hoping to publish this soon. We've already published the qPCR paper, but the bottom line is what you can see here is here's the CT value here. And here are a list of something like, I don't know, 15 or so samples. And what you can see is there's 6 dots for every sample. That's the report of everyone, their results of CT for a sample. And all of these dots are grouping together is what we're trying to show here. So they're pretty tight as far as how they reported their CT values across laboratories, both in the samples with high viral loads or low CTs, meaning there's a lot of virus in these samples, as well as in the low viral load samples when there's not much TLIV in the known positive and negative samples. So more variation, of course, when an assay is challenged at lower viral loads or higher CTs, but pretty tight around a CT of 20. So a summary of what we found is all laboratories reliably detected the negative and positive samples, except one laboratory reporting a single negative sample as a suspect positive, so right on the border of being suspect positive. So this interlaboratory validation trial provided data to support the 3rd stage of assay development as set forth by WOA for diagnostic assay validation. Okay, so that's kind of, I went through the disease. I talked a little bit about regulations and how we've responded as a country to this disease. And then I talked about some of the research that continues on development of assays for TLIV surveillance, such as the validation of ATACMAN qPCR. So in summary, I hope I've convinced you that TLIV is a global threat to the tilapia aquaculture industry. I hope I've convinced you of the importance of collaboration, that being private, university, and government labs, and how that led to the rapid detection of TLIV in the United States in 2019, following an import of Nile tilapia fingerlings from Thailand, and how USDA response prevented this disease from becoming established. And then once we knew it was potentially a risk coming into the United States, as we've been warning for some time, a risk assessment was quickly performed, and a federal order was put in place that again closed the door on tilapia coming into the country without an import health certificate, without having them looked at by a veterinarian in the exporting country and essentially having testing done in that country to ensure that they are negative for the disease before they ever get sent here. One of the problems that we encountered is, well, how do we do surveillance in the United States or even around the world? Well, we knew that we had a lack of validated TLIV diagnostic assays. In this day and age, qPCR is one of the fastest, most sensitive assays, readily used in diagnostic laboratories, not only in aquatics, but in terrestrial systems as well, human medicine or veterinary medicine. And so we designed this qPCR assay, as I described, and validated it stages 1 through 3 for WOA. And that assay came out, I guess that was the end of last year, I think. So that's available for folks. And finally, I wanted to mention some future directions on things. There's vaccine development going on, several groups within the United States. One that I was working with and continue to consult with and try to work with is Dr. Roy Curtis, who's developing an Wardziella piscicida DNA vaccine vector to introduce parts of the tilapia lake virus into tilapia to prevent infection by TLIV. There's lots of great collaborators on that, and that's work also being done by Dr. Kudi Subramaniam at the University of Florida and his colleagues there as well. We continue to work and understand the immunology and develop serologic tools for TLIV, and so this is Dr. Win Surochetpong, working with him, continuing to work with him We published about a year ago a tilapia, ELISA as well, and some important immunology work was actually in that paper as well. So those are kind of two important areas. We're starting to understand immunology better. The immunology informs vaccines and so on and so on. And several groups are working on breeding for genetic resistance as well, so that you have lines of tilapia that are resistant to tilapia lake virus. So that's future directions and such that is important and part of international collaborations and such. So I have a lot of people to acknowledge and thank. I wanted to make sure I acknowledge Bill Kelleher, again, that private laboratory, and the first person who was not only willing to do the diagnostic work, but send samples out for an unknown sample, which really protected the U.S. aquaculture industry, tilapia aquaculture industry. I mentioned Dr. Kudi Subramaniam, who is the Wildlife and Aquatic Veterinary Disease Laboratory, a very strong researcher and continues to do TLIV research. And then our collaborating laboratories for their interlaboratory trials and studies, Dr. Kevin Snethick at Waddle, our state laboratory, Dr. Reddy Bomanini, the Battle Laboratory, New Jersey Aquatic Animal Health Diagnostic Laboratory, Jan Lovey and his colleagues, and then USDA AFS. I have a lot of folks that were really critical to this study and making this whole thing a success, at least in my eyes. And from the trade standpoint, Dr. Shanna Siegel and Dr. Alicia Marston. From the commodity or domestic side on our home front, Nancy Hanaway, Kathleen Hartman, Lynn Creekmore. From an epi standpoint, we have Dr. Lori Gustafson and Christine Ellis. I don't think I said it, but Christine Ellis was the one who did the 2 risk assessments rapidly, which was critical. And then Janet Ward, who of course is a part of our National Veterinary Services Laboratory. All of these collaborations were critical to this story and are valued collaborators in this study. I'd like to leave you guys with just a few thoughts. This is for the youngsters, not the seasoned folks, but just a few things or thoughts that I have, the take homes perhaps of this work, food for thought, if you will. The importance of large scale collaborations, and for the younger folks to remember to communicate as well as you can and foster strong relationships. Something that my PhD advisor Ron Hedrick taught me long ago was stay on the merry-go-round. Which, what do I mean by that? It means that in collaborations, sometimes you'll get less than you deserve, and sometimes you'll get more, but just keep collaborating, keep remembering to be, have patience with people and continue to work with folks and, and just be patient with folks and collaborate and communicate. It's really, really, really critical to your success. No single paper is important to mess up collaborations. So remember that, think about the long term, and that collaboration is going to be important throughout the full term of your career, which could be 40 years. So think in terms of that, and remember the story that I told you today of TLIV. Without those strong collaborations, the work would have never been done. Perhaps TLIV would have become endemic in the United States, and so on and so on. And remember that researchers and diagnosticians impact U.S. aquaculture. What do I mean by that? Just remember that a lot of the work that I've done has been to find new viruses or describe new diseases. But the diseases that we focus on and the findings that we find and how we share them is important, but it impacts our aquaculture industry. And so, remember to report notifiable diseases. Remember that emerging diseases need to be reported, like tilapia lake virus. Again, Bill Kelleher from the private lab did not have to share that virus. And if he hadn't, this story wouldn't be able to be told, at least not in this manifestation. He actually was, in my opinion, brave to do that. And it did lead to issues, but issues that ultimately were the best outcome for the U.S. aquaculture industry. So, and it's also a reminder that, the diseases that we find really do impact our trade and regulations, okay? So now TLIV, before they can be shipped into before tilapia can be shipped into the United States, we've closed the door on bringing in TLIV infected animals through regulations. But, there's a cost to doing testing. So what you do will impact what you publish, what you report will impact U.S. aquaculture. Always think in terms of that. And working at USDA has helped me actually refocus and think more about the stakeholders and the impact of research that I've done, how it will impact aquaculture. So never lose sight of the industry and the commodities that we serve. I think that's another take-home message, at least one that's been important to my career. And then I wanted to also to mention that the aquatic animal health profession is dynamic and a wonderful path. So for those of you that might be young and looking for that path, there's many different avenues. I've walked in academia for 20 years and still walking in academia in many ways. I've now worked for the US government, but to remember, USDA, for those that might not have seen it, is hiring 5 or 6 new aquatic animal veterinarians or PhDs that are researchers that have subject matter expertise in aquatic animal health. It's, aquaculture is exploding. USDA is trying to meet that need from a trade perspective, from the domestic perspective, from the epi perspective. All of the units at USDA, if you like epidemiology, if you like diagnostics, if you like trade issues, there's all of the different units are hiring right now. So remember that it's not just academia that you can use your PhD, your master's, or your DVM with. You can work in government. It's not just USDA, right? The Fish and Wildlife Service has many veterinarians and PhDs. USGS, NOAA, that's just a little bit about government. There's so many opportunities as well at the state level, Department of Natural Resources, and so on and so on. And then finally, even private companies, something like Aqua Tactics out on the West Coast, on the East Coast, Kennebec River Biosciences, these private companies are hiring as well. So it's a wonderful career, and I wish everyone all success in that. And I hope some of the wisdom I imparted above can be helpful to you. Lastly, but not, I'd be happy to take any questions if there are any, and thanks so much for your attention. These are my greatest experiment, my 2 little wonderful darlings, Isabella and Lily, the loves of my life, the best part of my day. So thanks very much, and again, happy to take questions if there are any.
All right, thank you very much, Tom. That was a spectacular presentation, a very interesting story, and the advice there at the end was spot on. Don't consider myself young anymore, but it's still useful to hear.
Yeah.
All right, folks, feel free to drop your questions into the chat, or you can raise your hand or unmute yourself and ask Tom directly. And we'll start with one that came into me through the chat from Dave Groman. Dave's wondering if the USDA is setting up proficiency testing for labs that want to screen tilapia, either within the US or labs outside the US certifying imports.
Yeah, and I think I saw, I'll make a quick comment on it. It's not really my area, and I think I saw Janet is on the line. So Janet, if you wanted to make a comment on that, I wouldn't steal your thunder. But I'll make a comment if Janet's no longer available.
I am still on, Tom. Thank you. Dave, yes. The answer to that question is yes, that we will be offering check test panels for our AFS approved labs.
Awesome. Thanks, Janet.
We currently offer proficiency tests for spring viremia of carp, viral hemorrhagic septicemia, tilapia lake virus will be added to that, and infectious salmon anemia virus. And we've been starting to make some shrimp panels that eventually will be utilized in across our approved labs.
Great, thanks for the tag team there. Anybody want to unmute and ask the question? Yeah, go ahead. Kailash.
Hi, Tom. Are you somehow checking which assays are used by the laboratories outside US when they are checking for tilapia lake virus? And are you requesting certain assays and somehow controlling what they are doing during diagnostic to prevent from asymptomatic or persistently infected fish to come to US?
Yeah, so I can address part of that from the trade perspective. And I'll handle it from the USDA side and from a research side. So in the TACMAN qPCR paper, this relates to the analytic sensitivity, which goes straight to your question because you're talking about asymptomatic fish. I know what you are really meaning is that fish that have very low viral loads, right, that could slip through as being potential false negative animals, right? So you think they're negative, they look happy and healthy, but they might be carrying it, and so on and so on. So one of the things that we did was we looked at samples with very low viral loads, both as I described it with plasmid, but also in known infected samples as well as in the reproducibility study. And we showed again that it's very, very sensitive. No assay, as you know, is perfect, and a sample can slip by, or it can be in an organ you didn't test, and there's many ways that can happen. So there's no way to be absolutely sure that you are preventing diseases from making their way into the United States, of course. So that's an impossible thing, but it's a good question. And the other thing I would have mentioned from the research standpoint is we compared our assay to other assays as far as mismatches and the in silico performance and such. And so that goes to your other question about how other assays perform and such. And so we have looked at other assays, and there's a lot of mismatches among other assays and it's not because people didn't do a good job with those assays, the genomes weren't available, so they couldn't design better assays and such. So that's an important thing to remember as well. Do we know that there's a functionally significant effect of having lots of mismatches? In other words, other assays being used by other countries, do they or do they not do a good job? We don't know that. We haven't empirically tested that. I'll get to that at the end of this, but what we do know is you'd always use an assay with fewer mismatches if you have the choice. So we've thought about that, Nikolai. What USDA has done from a trade standpoint is there's a nested conventional PCR and a WOA or OIE disease card on the disease. That's the assay that we've asked people to use. That was prior to the qPCR being published and other assays coming out. And so that's the assay that's part of the Code of Federal Regulations at this point. And it's imperfect, but like any assay, but it does perform pretty well. And I think I can actually pass the baton again back to Janet here, but I'll just state that, and as far as your question about, has anyone been comparing assays, not only this assay, but other assays that have been developed in multiple laboratories, that has been done as a collaboration that I know the National Veterinary Services Laboratory and Janet was a part of. So I'm going to pass the baton. I'm glad Janet showed up today. Back to her, if you don't mind just speaking to at least your participation on that, Janet, that would be great.
So thanks, Tom. Yes, the National Veterinary Service Laboratories did participate in the round 2 of comparisons of PCR assays, those that are on the disease card and some newer ones that eventually will be published. So that is going on, and that there was a report that you can locate on the WOA on that round 2 comparison. And we've looked at that, we being us here at the NVSL, as well as Tom's assay on some direct, in addition to the panel from that comparison, but also from animals we've infected here with tilapia lake virus, and did time point testing of those over time. And that gets back to the discussions on, proficiency tests and check test panels. We will be distributing a standard operating procedure to our AFS approved labs, and that will be the assay that we will be utilizing. And then, as Tom May has indicated, that from what's coming into the country, those assays they're bilateral trade agreements that spell out which assays are used for that, and the health certificates or the health attestations that are required when animals come in with a certificate. So again, we're looking at all the assays so that we have, you know, the data to back up what we're requiring.
And just last thing on that, thank you, Janet. So Nikolai, I know exactly to the heart of your question. It's a really good question. Obviously, any assay can miss an asymptomatic carrier or a persistently infected low-level, animal. The Code of Federal Regulations at this point is suggesting, the nested conventional PCR. I have to look that up, to be honest, but it's the conventional PCR that's been around for a while. And that's imperfect, but that's the best that we have at this point. And it should be updated as these assays become part of the manual chapter of the World Organization for Animal Health, right? So the Aquatic Animal Manual. So one thing I didn't state is that TLIV is listed now, and a reference laboratory needs to be established. And when the manual chapter comes out, you'll have all the appropriate assays and their level of validation within that chapter. And it should list things like what Nikolai was actually asking, which is what level of validation, the analytic, diagnostic performance, reproducibility, all those kind of things. So that's a good question. And Nikolai, it's 7 o'clock or 8 o'clock or 9 o'clock. You shouldn't be working anymore, but I'm glad you joined. So good to see you, man.
Yeah, going home soon. Thanks.
All right, Tom, we got one last question for you here. This has been great. Where did it go? Did you, thinking about the pathways and the risk assessment, did you consider ornamental cichlids as a possible risk factor, and how did that shake out?
Yeah, so it's a great question. And no, that what was looked at that time predated some papers that came out in ornamental fishes. And so, regulations always fall behind the latest and greatest research. And so at the time, the known susceptible species were really those species that are susceptible, Nile tilapia, hybrid tilapia, red tilapia, and tilapia species at that point. And so that hadn't been looked at that point. So the regulations were really only related to the live tilapia industry. The question could be raised is, do ornamental species come in contact with our live tilapia aquaculture industry? I can speak to that very quickly. I've done some research on iridoviruses that are on the same farm, ornamental species with live Nile tilapia. So at least one other virus and different bacterial diseases, of course, easily transfer between ornamental and food fish, right? Many of these pathogens don't respect what industry they're in, and we are keeping ornamentals sometimes with food fish species. So is it a potential risk? Yes. Whether or not cichlids are, other cichlids, I should say, of course, right? The study that was done is on peacock cichlids, a Lake Malawi rift species, right? The Alanacara species of peacock. And so one of the things, so that study found that they could get disease. But one of the things that I wanted to make sure I mentioned about that is that WOA sets forth a series of standards that must be met before a species can be deemed susceptible and before regulations can actually be imparted on that species. And the way the study was done, which was not in any way flawed, but the way the study was done might not reach those standards. And so we don't know the risk, we don't know the host range of tilapia lake virus. And some of you may be privy to the fact that there's new viruses in this family being discovered. We found some in smallmouth bass, others have found them in guppies and things. And so this is going to be an area that's going to expand the family. We're going to find new viruses and so on and so on. But the host range of tilapia lake virus is not known at this point. And whether or not the new viruses that are being found are strains of TLIV or whether or not they're new species is important as well. So that relates to how WOA actually regulates things, how the ICTV calls species, so the International Committee on Taxonomy of Viruses, Will they call that variation that's being seen a new species because that impacts regulations? All of these things fit together. And I really do find it, I'll just make this last statement. I find it really fun to do research and diagnostics that impact our regulations and even sit on committees like the International Committee on Taxonomy of Viruses that, you know, makes decisions on what is a new species of virus and how that impacts trade. So a lot of this stuff all fits together, but that's a great question.
Well, that's great. Great. That's a great way to end it. And it's clear the story is going to continue. So we'll have you back in a couple of years to tell us part 2.
Thanks.
Thanks very much, Tom. We appreciate your presentation today and taking the time to talk to us.